Since the initial outbreak of the coronavirus, many patients have reported enduring neurological issues, such as brain fog, fatigue, and memory loss. To understand what happens in the central nervous system, researchers recently reviewed 49 brain imaging studies that compared the MRI scans of people who had contracted COVID-19 with those of healthy individuals. The review found that a COVID-19 infection was associated with widespread structural and functional differences in the brain. When looking at brain structure, the researchers found consistent patterns of abnormalities across several key regions. Patients often showed a reduction in overall gray matter volume and a thinning of the outer cortical layer. The frontal lobe, which handles decision-making and cognitive control, frequently exhibited structural changes. Differences were also noted in the temporal and parietal lobes, areas involved in sensory processing and attention. These structural shifts might help explain why some recovering patients experience difficulties with top-down cognitive control, visual attention, and auditory processing. The review also highlighted extensive issues in the brain's white matter, which contains the nerve fibers connecting different processing centers. By tracking the microscopic diffusion of water molecules, scientists found abnormal water movement in COVID-19 patients. This indicates that the protective coating around the nerve fibers had degraded. These microscopic changes were present even in patients who experienced only mild to moderate respiratory symptoms. Functional imaging scans, which measure blood flow or oxygen levels, revealed altered patterns of brain activity. When patients were simply resting in the scanner, their brains showed abnormal synchronization between different regions. The limbic system, a network of deep brain structures that regulates emotion and memory, frequently exhibited connectivity issues. These functional disruptions correlated with clinical symptoms like anxiety, depression, and post-traumatic stress. Additionally, tests measuring cerebral blood flow found lower than normal circulation in several brain areas, including deep relay centers like the thalamus. The researchers note a key limitation in the reviewed studies. They rely heavily on cross-sectional data, capturing a single snapshot of the brain after infection. Because the vast majority of these studies lack pre-infection baseline brain scans, it is difficult to prove definitively that COVID-19 directly caused all the observed changes. Individual biological differences present before the pandemic might account for some of the variations in brain structure and function. Future research with long-term tracking will be needed to see if these neurological changes are permanent or reversible over time. Learn more about this study and other new research at PsyPost.org.